My name is Mary Stenzel-Poore. I'm a researcher from Oregon Health Sciences University. Today I'm going to tell you about my team's work on endogenous neuroprotective mechanisms that can protect the brain from ischemic injury. Imagine that over the next 10 minutes, 10 individuals in this country will have a stroke. And these events cannot be anticipated. However, over the same 10-minute period, there will be 10 individuals who have a cardiovascular procedure, which puts them at significant risk for an ischemic event to the brain. And these procedures and these events can be anticipated. What I would like to tell you about today is the work that my team is doing to help develop a prophylactic strategy, a strategy that you could give in advance to protect the brain of an individual that is going to have one of those cardiovascular procedures and spare them the devastating effect of ischemic brain injury. So in order to do that, I need to explain how we can harness a very powerful protective mechanism that the brain has against injury. And that protective mechanism is induced by a phenomenon called preconditioning. And preconditioning is a stimulus that, when given at a very low dose, induces profound protection against subsequent injury. It's essentially the idea that what doesn't kill you makes you stronger. And in this, we think of a variety of stimuli that in large dose would be injurious, but in very small dose actually provides protection. And examples are hypothermia is an example, volatile anesthetics, another example. Actually, very brief ischemia is another example. And then there are receptors of the innate immune system, toll-like receptors, that actually can be stimulated with agonists for toll-like receptors that are able in very small dose to give you profound protection in the brain, in the kidney, in the lung, in the liver. And these receptors are on many cell types, cells of the immune system, cells in the brain, cells on the endothelium that line the vessels. So in our system, we work with one of the toll receptors, toll-9, and we have developed a drug that you can give to an animal, and we use rodents as our model. We've developed a rodent model of stroke that we can give a little bit of this toll-9 agonist. These are small pieces of DNA that are methylated. And when we do this, we can give the animal significant protection. In fact, the stroke that animal has, if it's been given this drug in advance, is about half the size of normal. So we said, how can we determine what it is the brain is doing itself to protect the animal? And the way we went about determining this is we took different forms of preconditioning. We used ischemia, brief ischemia, and we used 2 different toll receptor agonists. And we compared each one of them with the idea in mind that if you could imagine comparing each one of them, that there might be a convergent pathway that actually was the core pathway that provided protection to the animal. And we did that experiment. We compared endotoxin tolerance, or giving a little bit of lipopolysaccharide, or a small dose of CpG DNA, or brief ischemia. And what they all had in common were 12 genes that you saw only in those animals that had been preconditioned prior to their stroke. And those 12 genes all link to an interferon pathway. So that was a brand new pathway involved in protecting the brain. So what we learned from that is that preconditioning induces a reprogramming of the cell in response to injury. And they induce the cells in response to injury now turn on a profoundly protective mechanism. So how do we begin to translate that whole idea and this notion of being able to protect the brain in advance of at-risk patients who would have a ischemic event during a cardiovascular procedure? What's the translation pathway look like? How do we get from bench to bedside on that? Well, we decided to develop a non-human primate model that would allow us to really be able to model the injury in a very relevant species, a species that closely mimics humans. Why did we do that? Well, we did that because in the stroke field, we have had many failures of drugs that we found to be very protective in our rodent models that simply didn't translate to humans. And my laboratory decided to develop the first model for stroke in the rhesus macaque. We've now tested our drug in these animals, and we have found profound protection. We can give low doses of a Toll-9 agonist and then give these animals a brief or long period of ischemia. And these animals show incredible resistance to the injury. So we found that this is a powerful neuroprotectant. The animals live and can take care of themselves. They have about half the brain injury that an animal who's not so treated demonstrates. So the conclusion from this is that the brain's own endogenous mechanism of protection can be harnessed and used to protect at-risk population. And so we are now to the point of wanting to take this into a phase 1, 2, 3 clinical trial that will allow us to truly test whether or not patients who are going to have a cardiovascular procedure such as coronary artery bypass or carotid stenting or an endarterectomy, all procedures that put you at risk of having embolic events in the brain. We would like to now take this drug, which is a very safe drug, and take it into patients to really test whether this can work. So what I think I've told you about today is I've told you about our fascinating studies to really start to understand what are the mechanisms that preconditioning employs in order to protect the brain. And I've shown you what I believe is a proof of principle where we've been able to test this drug and show that it can protect the non-human primate brain, a very close model of stroke in humans. And I hope this brings also to many the promise of this kind of therapy, of this kind of approach to protecting against a number of different kinds of insults to the brain or to other organs. Thank you for your time.